nodes, and cell differentiation. For this model of network formation to work, the precise shape and sequence of the three-dimensional RNA flags are critical. RNA Pol II elongation pauses cannot be random if gene-to-gene contact is to be precise. During embryonic development, the number of paused RNA Pol IIs increase, their regulation determined within each cell type. It is not known what defines a pause point, but if pause points were precisely regulated during differentiation, the resulting network of linked genes would be precisely regulated. For example, here's a mother cell and some important gene. Say it's a stem cell containing a gene required for differentiation. It divides. The same gene is present in both daughter cells, but in the first daughter cell, the RNA Pol II pauses consistently here, and I will discuss how this might happen in the next segment. And in the second daughter cell, it pauses there. Why does this matter? Well, in my model, a pause leads to the release of specific flags that signal to a separate cluster of genes. When flag blue is released, then boom, the RNA Pol IIs on genes X, Y, and Z are also released. These genes might code for proteins that would ultimately make the cell a muscle cell. But suppose in the second daughter cell, that pause site is hidden. And again, I'll talk about how this might happen in the next segment. And now the RNA Pol II pauses further down the line. A different flag would be sent out, and genes Q, R, and S might be turned on. These daughter cells have been started on a different path of differentiation, perhaps into neurons. With this model, it is now easy to imagine that a loss of a critical pause point or a central node could lead to loss of cell control and even cancer.